So, good afternoon, everybody. You have the stragglers. So, I'm Scott Ripley. I have the pleasure at Cytiva of leading two business units, one focused on nucleic acid therapeutics, the other focused on drug product formulation with precision nanosystems. This afternoon, Science Slam, we're going to focus on what I think is the very exciting field of the role that mRNA can play in enabling the next wave of immuno-oncology based therapies. We've got three speakers this afternoon, which I'll introduce in short order, and then we're going to have a little bit of a moderated session. But we've changed around the order a little bit. First up is going to be Sadik Kassim from Danaher, who's going to give us a little bit of a broader overview of some of the approaches being taken to immuno-oncology. Next is going to be Adrian of Capstan Therapeutics, who's going to talk to us about in vivo approaches to immuno-oncology. And then we're going to round out with Geall from Replicate Biosciences and the approach that they're taking to tackle immuno-oncology. So it's a pleasure to welcome up Sadik Kassim.
Great, good afternoon. Thanks, Scott, for that introduction. Thanks to the organizers for the opportunity to present. And I look forward to slamming some science in your face this afternoon. Science slam. All right. So mRNA, I think really the attraction of mRNA is it's in its flexibility as a modality. It touches on different applications. So what this slide highlights is that the initial application that all of us are familiar with is in the vaccine space. But beyond vaccines, within the immuno-oncology space, mRNA can be used to reprogram cells as an alternative to viral vector, gene modification to retarget immune cells towards cancer. It can be used to deliver monoclonal antibodies, cytokines, and chemokines that can activate the immune response and really turn a cold tumor hot. It can be used as a personalized cancer platform, and it can be used as a adjuvant to augment the innate immune response. So I think each of these applications are very exciting, but each of these applications requires a very specific set of considerations to really translate into the clinic. And what I want to do here in this talk over the next few minutes is highlight some of the outstanding gaps in the field to enable the translation, and also highlighting some of the considerations when developing these into drug products. So I think the first consideration with regard to the final use case is really at the sequence level. So one of the attractive features of mRNA, it's a modular molecule. So it's composed of the cap on one side. Oh, yeah, if we can go back 2 slides. So it's composed of the cap, which determines the stability and the translatability of the molecule. Then you have the open reading frame, which encodes the gene of interest. Then you have the UTRs, untranslated regions, which determine things like stability and, in certain cases, tissue tropism. And so modifying these different regions when designing the sequence can have an impact on the half-life of the mRNA molecule, can have an impact on the translation, meaning the dose of the mRNA molecule. So I think that modular flexibility enables the use cases that I highlighted in the beginning, but the design rules for the sequences still remain outstanding. So for example, although we have the protein structures for over 200 million proteins via things like AlphaFold, I think our understanding of basic things like secondary structure for mRNA and the relationship between sequence and function and sequence and structure remains really very minimal and in early stages. The second consideration when designing mRNA as a drug product is really the scale and the application. So all of us are familiar with mRNA and its success as a pandemic level response for COVID. The scale there very small doses, micrograms per patient, and billions of doses globally. And the logistics from a manufacturing perspective are reaction volumes of up to 100 liters of IVT, and really in the kilograms of production of drug product. In the intermediate applications, so like cytokine replacement or replacement for monoclonal antibodies, the scale and the architecture becomes a little smaller and manageable. So I think the clean room footprint becomes fairly small and easily established even by a biotech or an academic facility. There we're talking about scales, reaction volumes of 0.1 to 10 liters and doses of 1 gram to 50 kilograms. But the ultimate use case where you require flexibility is in the personalized cancer vaccine space. So here we're talking about bespoke, one dose at a time, specific and unique to the patient, micrograms, reaction volumes in the 10 milliliters to 50 milliliters per scale, and production per year of less than a gram. So the question becomes, from a consideration and a manufacturing infrastructure perspective, how do you actually develop that infrastructure to enable translation of something like a personalized therapy? And I think there's a lot of interaction between these different scales. So the infrastructure that's developed for personalized therapies will enable advancements at the intermediate and large scale, and vice versa. So one thing that I wanted to highlight here is the recent cancer data from BioNTech, Pfizer, very similar results from Mrna, highlighting the production benchmarks and the unique considerations one needs to take into consideration when designing a personalized mRNA vaccine. So from the initial tumor biopsy that was used to identify the neoantigens and then design the mRNA, all the way to the final drug product manufacturing and release, it took BioNTech and Roche less than 9 weeks. So less than 5 minutes to transport the specimen to pathology, to then fix and really prepare for sequencing, then to ship to BioNTech for the identification through the algorithms of the dominant neoantigens, and then the production and the release of the QC for that specific patient in less than 9 weeks. So these are logistics and infrastructure that we don't currently have in place. We have pieces across the ecosystem. Within Danaher, the one way that we're thinking about it is working through our family of companies, IDT and Archer. We can help with that initial NGS library prep and sequencing, and then that gets translated into donor templates that can then be made into plasmid DNA or synthetic DNA that can be used as the template for IVT mRNA for small reactions, and then formulation, fill and finish, and analytical characterization. So from a personalized perspective, really it's putting together these capabilities end to end and enabling it. And within the Danaher family, if you're a drug developer and you want to build this internally, you can go through the Cytiva family of companies, or if you want to do this externally, we had a really great talk yesterday from Todd at Aldebaran talking about the end-to-end mRNA sequencing capability and end-to-end mRNA drug product manufacturing. Now, an additional consideration beyond sequencing and manufacturing at small microscales is really the design and manufacturing of delivery vehicles. So the first generation of clinically validated lipid nanoparticles have really demonstrated great success in the delivery of siRNA and mRNA in small doses with the vaccine. But really the future, if we're going to target selectively tumor tissue, or we're going to target selectively the reprogramming of immune cells within the tumor microcontexture, we need to develop next generation particles that can deliver mRNA in a very selective, targeted, and specific manner. And the design space here is really large and remains largely unexplored. So lipid nanoparticles, although have demonstrated clinical success, there's a lot of success, especially with chemotherapeutic reagents, of polymeric nanoparticles. So can can we leverage that insight from polymeric nanoparticles to generate next-generation immuno-oncology therapies using mRNA? Likewise, there are hybrid solutions that are being developed, and I think beyond the chemistry of these delivery vehicles, the other opportunity really relates to the conjugation and the functionalization of the nanoparticle so that it can target specifically the tissue of interest. But also, from a size and architecture perspective, we know that the size and the shape of the nanoparticle has an impact on the biodistribution and the tissue tropism and specificity of the nanoparticle. And these are all factors that haven't really been explored and really need to be considered when designing an mRNA therapeutic for a personalized vaccine or an antibody replacement or in other applications for immuno-oncology. The final thing to take into consideration is oncology is difficult, and what we're realizing in the field of immuno-oncology that it's not going to be a single solution that's going to potentially treat the cancer, but it's really going to be combination therapies. So I think the first published reports from Mrna and BioNTech and Roche have demonstrated that personalized mRNA therapeutic vaccines work really effectively in combination with anti-PD-1s, Keytruda, the BMS Opdivo programs. And likewise, in the broader immuno-oncology space, we're seeing really great proof of concept from various companies mixing and matching different therapeutic modalities. So I think the treatment paradigm, and something to take into consider, depending on the indication and the histology that you're targeting, really mRNA is not going to be the catch-all and be-all, but it's going to be in combination with other things, which brings in a lot of preclinical and manufacturing and clinical development complexity. So to conclude, I think within the context of Danaher, the way that we think of it is that the field is still fluid and it's evolving, and our family of companies are really positioning themselves to work with drug developers to really customize and develop specific solutions, whether it's antibody replacement, personalized vaccines, whether it's insourced or outsourced. So I would encourage you to talk to some of the folks here, some of my colleagues that are in attendance. Thank you.
Okay. Let me start by thanking Scott first for the invitation to be on this panel. Let's get going. That may not be my presentation, that's replicate. If you can, I can go to replicate presentation, then finish with Capstan. So I'm going to talk today about how we are thinking in terms of utilizing mRNA as a very versatile tool in order to generate next wave of transformative treatments for cancer and beyond. I'll focus on cancer today, but we have interests broader than just oncology. And let me just start with reminding everyone some of the major pillars of our therapeutic armamentarium in cancer, and especially focusing your attention on the latest developments. 6 products approved to date. We are celebrating, of course, Kymriah and Yescarta, 6th-year anniversary this year. But the question is, what's next? What are those tools that we need to create and leverage in order to really scale up, scale out the concept of immunotherapy and unleash its full potential? There are 2 major questions in oncology arena that we are facing. Number one, how do we consolidate patients into remission and long, durable response, complete remission, cure, eventually in frontline, first-line setting? So that's the first question. And the second one, how do we need to rethink the approach for solid tumors? That's a formidable challenge that we still face. And the way we are thinking about it is in vivo reprogramming of the immune system. In vivo reprogramming can be done with a variety of tools. The one that is scalable, as in very interesting to us, are targeted lipid nanoparticles depicted here on the left-hand part of the slide. And we also needed to rethink and optimize that particular tool for delivering mRNAs that encode, for example, chimeric antigen receptors or biological response modifiers. Number one, the ionizable lipids that we need to use need to fit the purpose. Vaccine-type ionizable lipids are no longer safe tolerated if we want to deliver large amounts of mRNA or RNA cargo in general. And also, we need to rethink the selectivity in terms of delivery at cell level. We need to be able to reprogram T cells, subsets of T cell, or other immune effector cells, and diminish, mitigate, detune liver as well as other tissues. And that's how it works in principle on the right-hand part of the slide. You see that these targeted lipid nanoparticles can be taken up very rapidly, actually, in a receptor binder mediated fashion into the endolysosomes of immune cells, including T cells. And the ionizable lipid within the endolysosomal compartment is initiating the release of the payload into the cytoplasm through an endolysosomal membrane breach. The chimeric antigen receptor is expressed very rapidly within hours. We are skipping transcription. This is translated directly into polypeptide, redirecting the T cells in situ against pathogenic cells, be those cancer cells or other types of cells. So that's how it works based on the experience to date that we have. So mRNA provides us with the versatility to tune up and down the AUC, the exposure. We have more control. We can actually adjust the exposure that we need to the high therapeutic safety bar that actually needs to be met in minimal residual disease or frontline setting or outpatient community, or actually multiplex various mRNA payloads, dial in biological response modifiers, more antigen receptors, CARs and TCRs, in order to attack a solid tumor process from multiple angles. We have a great deal of data that we and others published to date in context of developing first wave ex vivo engineered CAR T cells that, of course, rely on retroviral or lentiviral vectors, which is one disadvantage when you are thinking about scaling up and scaling out this concept. But this data, and we show here axi-cell and third-line DLBCL, work that we published along the last 5 years. These data were an excellent springboard for us to develop the metrics that we needed to follow and to guide the optimization of in vivo reprogramming approaches. And that's how we anticipate this will work. It will work through one or multiple administrations of the mRNA-loaded targeted LNPs that will be needed to emulate an area under the curve that is necessary for achieving a response or a durable response in a setting such as diffuse large B-cell lymphoma. An advantage, of course, of having this type of treatment is that we do not need lymphodepletion conditioning, but we can co-opt the endogenous immunity. And just refreshing your memory in terms of the type of data that we published in DLBCL before, third line, about 40% patients achieving durable response and potential cure. More importantly, moving this treatment modality to second line and first line, we showed that the objective response rate, complete response rate increased, presumably due to the fact that you have increased T-cell fitness, as well as a more favorable tumor microenvironment. So clearly, this speaks to the importance of moving eventually this treatment modality in frontline setting, first line, in order to aspire for cure. And you can multiplex also mRNAs that target different antigens that are complementary to each other in terms of expression profiles, such as CD19 and CD20 in diffuse large B-cell lymphoma, or CD19 BCMA in multiple myeloma, and I can go on and on. So I tried in one single slide to provide you with our thinking in terms of how RNA therapeutics can be utilized for in vivo reprogramming of the immune system, in general cells and tissues. On the left-hand part of the slide, you see the advantages for this technology in oncology. And of course, in oncology, we face a higher efficacy bar, but a lower safety bar. So they're multiplexing a variety of elements, including, for example, biological response modifiers, such as gamma chain receptor cytokines, immune checkpoint blocking agents, together with engrafting novel antigen receptors, regenerating a repertoire, could be a formidable way to attack the cancerous process even in later stages of development, something that has not been done yet. So in terms of other indications, non-oncology areas such as autoimmunity, regenerative medicine, fibrosis, and so on and so forth, the versatility of mRNA is such that it provides us with a tunable approach to actually diminish the exposure, just sufficient to be efficacious, but meeting the very high safety bar that we cannot meet with the current generation of ex vivo viral engineered CAR T-cell interventions. So I'm going to just complete with a picture of how we are thinking about bringing this technology eventually to treatment of solid tumors. It's going to be a formidable challenge. It's going to be much more difficult than our previous work with B-cell malignancies and ex vivo engineered CAR T-cells. But the way we are thinking about solid tumors is through integrated attack from three major directions. And all of this can be, in principle, achievable with mRNA-based technologies that are aimed to, number one, regenerate, restore, or create an immune repertoire against tumor-associated antigens. Number two, expand and mobilize a variety of arms, broader arms of the immune system, including innate immunity, myeloid cells, NK cells, in addition to T cells. And number three, overcome detrimental immune inhibiting mechanisms. For example, the stroma, which has a very deleterious mechanistic effect, as well as immune checkpoints that have been described to date. With this, I would like to thank you very much for the attention and welcome the next speaker.
I'm so happy you didn't present my slides. Nice to meet everybody. For those of you who don't know me, my name is Andy Geall. I'm Chief Development Officer at Replicate Biosciences. We're based here in Sorrento Valley in San Diego. We're one of the second generation mRNA companies that are starting to emerge. We're taking advantage of the manufacturing delivery solutions that are out there and focusing in on biology. We focus in on, there we go, self-replicating RNA. These are much larger than conventional linear RNA. A COVID vaccine, for instance, is around 4,500 to 5,000 bases. Our RNA is somewhere from 9,000 to 16,000 bases. We encode viral replicase that allows self-amplification or replication of the RNA, and we get much higher levels of protein expression. We've managed to unlock the zoo of alpha viruses. So we have 15 novel vectors with 15 sets of novel non-structural proteins that have a diverse set of biology. What that allows us to do is develop vectors that are good for vaccine applications where we're looking to generate potent antibody responses or potent antibody and T-cell responses. One of our focuses is oncology, and we're trying to derive very potent CD8 T-cell responses. But then we also have vectors that seem to be non-engaging in the immune system and are good for just pure probably gene therapy type applications where you're looking to drive protein expression. And we're probably about 1,000-fold above what you're currently seeing for linear mRNA. So it's a very exciting and dynamic space. So we were co-founded in 2020. So we're a very new company. We're still on our Series A funding. We're clinical. We'll have our first program we'll read out towards the end of the year. It's a second generation RNA vaccine approach in rabies. We're hoping to see seroconversion at very low doses, 0.11 microgram, at scales of manufacture that people haven't seen before. If we seroconvert a 0.1 microgram, we can produce 50 million human doses per liter in 2 hours. So a significant advancement in what we've seen, but we get not only antibody responses, but T-cell responses. So we'll report those data shortly towards the end of the year. But As those T-cell responses read out, we're then going to move our second programs into oncology, where we think we can have an impact on things like metastatic breast cancer. And the third vision is then, as we learn more, is to start rolling out the technology into cell therapy and gene therapy. So we see the landscape of, we're very excited about the current state of the COVID vaccines, but we really see these as a hit and run application. It's a short burst of protein expression and a short burst of immunogenicity that then drops off. We're looking to extend the burst of that protein expression, extend the durability of immunogenicity, and drop the doses significantly lower. Doing that, we'll start to come under the adverse event profile and have vaccines that are much better tolerated, and hopefully with a magnitude of immune response that we're not currently seeing, and a durability that we're not seeing. We're starting to see stretch applications from companies like Pfizer, Moderna, and biotech in multivalent vaccines, personalized cancer vaccines. But to really extend the technology beyond that, we've got to see a significant improvement in the amount of protein produced and the duration of expression. And we feel that the second generation self-replicating RNA systems will achieve that. So this is our strategy. We have built out this novel library of vectors with a diverse set of biology that we're going to roll out. The first target is in infectious disease. We've established manufacturing capabilities within the CDMO network. It's taken us a few years to work with a company in Massachusetts called Curia to manufacture large RNA, and currently we're manufacturing in the size range of 9 to 16 kb, but it's established, it's the same raw materials going in. We've just adapted the process to be more amenable to these more unstable large RNAs. We're formulating our material at Precision Nanosystems in Vancouver. We've taken a license into their lipid library. We don't see the need to build that capability out ourselves. We're focused on the biology of the vector, and we've successfully filed an IND, and we're dosing patients using that strategy. Once we see a readout from that clinical trial, it'll be both neutralizing antibodies, which is relevant for rabies, but we'll also be measuring T cells. If we see potent T cells, we can then bring the technology into the oncology space, and our first targets will be things like metastatic breast cancer. During first-line therapy, treatment through a hormonal receptor leads to a known set of mutations. We're going to encode those antigen mutations in our vector. A 16 kb vector allows us to code a lot of antigens. And so as single tumor cells start to mutate during frontline therapy, T cells will recognize those cells and then cleave them out, and we'll be able to keep the tumor under suppression of frontline therapy. If that strategy works, we'll then be able to look at other targets like lung cancer and prostate cancer, where known mutation pathways are already established. And then finally, we're looking to move into more of the immunology space, where we're trying to target the inflammasome and downplay autoimmunity. So here's our strategy. In infectious disease first, we're already in patients. We'll have the readout towards the end of the year. Hopefully the T cell responses are potent, and then we'll roll into our oncology programs. And then finally, we're already starting to look at the immunology space. We're encoding cytokines, chemokines, particularly proteins with very, very short half-lives in the 1 to 2 hours. We can express them for several weeks, so a significant advantage. So with that, I'll say thank you and take any questions as the panel comes up.
Thank you, Andy. If I can ask the other panel members to join us on stage. Thank you. So I think we'll have some time towards the end for questions to the audience. So certainly if you do have questions, be thinking through those. But hopefully you got a little bit of a flavor there of the numerous different approaches that have been taken for immuno-oncology leveraging mRNA. I guess, Adrian, a question to you. By my own analysis, I've seen at least 7 fundamentally different approaches to tackling immuno-oncology. Sadik talked about whether it's cancer vaccines, whether it's in vivo cell therapy. Could you maybe expand a little bit on the number of different approaches and end molecules that you're seeing tackled with mRNA in the immuno-oncology space?
No, thank you very much for the question. I think it's great to think more strategically indeed and to learn from actually the trials that have been done in the past with a variety of different therapeutic modalities. But I feel very strong that based on all the data that we generated to date, as well as others, we need to tackle from 3 different angles the problem of oncology. And I think that mRNA, RNA in general, is an extremely versatile tool that will allow us to do that eventually. So number 1, transferring antigen receptors in vivo eventually, both chimeric antigen receptors and or T-cell receptors, because the intracellular proteome is extremely important, especially in solid tumors from an immunology standpoint. Number 2, payloads that are biological response modifiers. I feel very strong about those. I think we can do a lot in terms of rejuvenating the immune system, the T-cell fitness, so to speak, directly in vivo by transferring gamma chain receptor cytokines or other biological response modifiers through mRNA. And at last but not least, another big category is that of immune checkpoints. We need to be able to overcome all the barriers in front of concerted immunological attack against the tumor. So expressing in the tumor microenvironment immune checkpoints, co-targeting the stroma, which has a highly deleterious effect on the immune system in the microenvironment, I think is a very important aspect. And we need to be able to tackle all these 3 major directions simultaneously in a global, rational way, hopefully in vivo. Thank you for the question.
Perfect. Thank you. Andy, you shared the approach of replicate with the self-replicating RNA, the 2nd generation. I mean, I think just fundamentally the sheer potency of those molecules moving on from the 1st generation gets interesting. But I think you also shared how replicate strategy is to start first with rabies. I know you're entering sort of human trials right now.
We're in trials.
But you talked about the approach to immuno-oncology. Help us understand how mRNA almost acts as a platform within replicate to allow you to step from an infectious disease approach to an immuno-oncology approach, and how that helps accelerate what you might think about in immuno-oncology.
Yeah, I mean, the first thing that attracted me back in 2008 into mRNA was the production process. So it's a cell-free enzymatic production process. I worked at Novartis at the time. We had, sites that were billion-dollar facilities, and they made a single protein-based vaccine. And so for the first time, we had the ability to scale down the technology and produce doses at in a bioreactor at levels we'd never seen. But the process of making RNA is generic, and you can encode pretty much anything you want. So Sadik had a list up there. They're all doable, but the manufacturing process is the same, and the speed now to get from point A to point B is very fast. Replica is on a Series A. We'll have a finished phase 1 clinical trial by the end of the year, and a second one going in. That's unheard of, before. You'd be on your C, your D, you'd be struggling for financing. And so we're starting to see an acceleration of research and development and clinical application like we've never seen before.
Absolutely. I mean, you paint the picture there of that sort of speed, but Sadik, when you opened up, it's fascinating that this market, and my background came from a sort of bioprocessing space, and I've never seen a market start in reverse with perhaps the biggest process there will ever be, which is the COVID vaccine, sort of vaccinate the planet. But on the other end of the spectrum, Sadik, you were introducing the concept of personalized cancer vaccines, vaccinate the individual. So you've got a spectrum of scales there that sort of boggles the mind. Can you expand a little bit maybe on what Andy was saying about how that has implications for how we think about CMC approaches and what that means for future sort of manufacturing infrastructure considerations or considerations for mRNA LNP overall?
Yeah, absolutely. I mean, I think one of the advantages with large-scale CMC footprints is that you have centralized QC, and that really enables consistent quality control and production of the manufacturing. With this architecture, now you have the ability to decentralize that capability and really have it in a very small footprint, printer size. And I think the manufacturing is one thing that is going to enable innovation in the personalized space. But I think the thing that we're lacking is really that parallel analytical development and release that can really quantify what it is that you're making at small scale, the quality of it, and really release it in real time. So I think right now we're sort of at this really, thoughtful innovation space where we have the, to Andy's point, like the process is generic, like whether it's large scale or small scale. And there's some small variables like the T7 enzyme that you use, the capping method that you use. Is it enzymatic? Is it co-capping? I mean, those are all, I think minor things, but I think the thing that we still need to wrap our heads around is really, as we get those bigger payloads, I mean, as you're talking about self-amplifying, self-replicating RNA, then we have circular RNA. Are the analytical methods catching up to really enable that, acceleration of development translationally and clinically, whether it's small scale or large scale?
Perfect, thank you. A question maybe I'll direct to both Andy and Adrian. You talked about some of the efficacy there. So the self-replicating, Andy, you positioned it as sort of a second generation RNA. What are some of the approaches that are being taken to focus on efficacy, whether it is payload expression, payload targeting, that are really relevant to the immuno-oncology space at the moment? Maybe start with Adrian and then.
We have a great deal of experience from our prior efforts with chimeric antigen receptors in terms of how long do we need to express a chimeric antigen receptor in vivo, ex vivo, and all of that, and especially what's the level of expression that you need to attain in order to maintain that polyfunctionality, right? So if you rethink that and try to translate the concept from retro and lentiviral vectors that we use for the first wave CARs to mRNA, you're dealing with transient CAR expression, but the beauty of it is the tunability. You can tune up and down your exposure depending on the therapeutic efficacy and safety bar. So I think for mRNA, it's going to be very important, and in general for chimeric antigen receptors, TCR products, to rethink the architecture of the construct, as well as the mRNA itself, even non-coding areas such as UTRs, right? In order to codon optimization, another example, in order to drive up that expression level and achieve sufficient persistence for what we call a serial killing effect in vivo. So through these approaches, you can tune up and down the strength of your effector cells, as well as achieve that exposure that is needed for a higher or a therapeutic efficacy bar or a higher safety bar, tune down the exposure if that's needed. Now, autoimmunity, regenerative medicine, and fibrosis.
Yeah, I'm going to, Scott, talk about delivery. So for those of you who don't know the history of mRNA vaccines, my team at Novartis was actually the first to test an LNP RNA. We published a paper in 2012 in PNS. My name was the first author. But we've been exploring them since 2008. We'd seen them as a great tool to deliver a vaccine. In this case, it was RNA. We had a big collaboration with Alnylam. They were doing systemic delivery and really on the edge of therapeutic efficacy and toxicity. And we thought we could enable a strategy with an RNA, and it worked pretty much straight away. We didn't develop it. It's coming out of the Peter Cullis lab. But over the next 7 years, we screened hundreds of LNPs to look to see how delivery impacted, the way the RNA interacted with the human body. And some LNPs were really good at vaccine applications. Some were tuned down a little bit and capable of much higher levels of protein expression. What we're starting to see with these much more potent vectors now is the ability to polymers don't traffic to the immune compartment the way LNPs do. All LNPs seem to tickle the immune system, and there's a bit of a therapeutic window of opportunity, but once you're above it, you start to see adverse events, and particularly if you're going to have to do multiple dosing. So we think for the first time we're at a point with polymer delivery with these vectors that we can open up the gene therapy space and start to express protein without hitting, an immune response against that protein.
Thank you. Thank you. A big part of the theme of the whole conference really is how we bring some of these life-transforming therapies to the patients, ultimately. So there's some fantastic science, but unless it's getting to the patient, it's sort of, I guess maybe to the whole panel, could you, Adrian, you're talking about in vivo cell therapies, which of course by extension is hugely compressing that workflow, has an impact on sort of the patient experience, delivery to the patient. Andy, you talked about vanishingly small doses and what that might mean. So how do we think about some of the potential advantages or advances in mRNA, LNP that might help accelerate the uptake of this in the healthcare system and the patient experience as well?
Do you want me to take a stab first?
Yeah,
or
sure.
Sadik?
Yeah.
So I'm thinking that we have to work on two different levels. Number one, access, scalability, access, being able to scale up the technology in order to service patients in an outpatient setting, ambulatory setting, and earlier line therapy in general, as well as non-oncology indications. So safety is very important, as well as scalability. COGS are very important as well. But from a safety standpoint, if you're talking about TLNP or LNP mRNA category, then the ionizable lipid, the choice of ionizable lipid will be extremely critical, because the ionizable lipid is driving, at least based on the emerging data, the safety profile, especially in context of redosing. So we have to think that aspect of the business. The second aspect is, of course, the mRNA itself, and solutions that are offered by various companies, and our speakers actually today went through those in terms of how do you scale the mRNA to be able to service not only clinical trials, but in commercial setting, millions and millions of patients. That's a key question. And clinical performance is another important aspect. You have to be able to show that you have the therapeutic index and the safety margin that fits us or is suitable for a certain disease indication. So in non-oncology area, what we are facing is a very high safety bar. So we need to understand how we can tune up and down this exposure. How can we actually stay away, in context of immunotherapy, from CRS or ICANS, neurotoxicity grade 1 and above. Otherwise, we'll need to hospitalize the patients. And at last but not least, the efficacy aspect is extremely important as well. So in autoimmunity, regenerative medicine, we may not need to dial in features such as multiplexing. We may be able to develop product concepts with an easier, so to speak, target product profile. But in solid tumors, it's going to be different. I'm going to pause here.
Andy?
Yeah, I mean, we saw implementation of COVID vaccines in a, emergency response setting. The regulators have dialed back that now. So the level of scrutiny is now changed. We'll see acceleration into the clinic and approval in those unmet needs where, the things can be tolerated, but we're going to see things slowing down dramatically. Prophylactic vaccines in healthy patient populations, they're going to want to see huge, swaths of safety data over extended periods of time. The same they've asked for traditional vaccines. And so it depends where you go and how the technology is applied and the setting and the risk, profile that you're going into. So it'll be a mixed bag. Some things will go very fast in oncology, but outside, as we step back and the safety criteria get more and more extensive, it's going to get harder and harder, and it's going to take time, and sometimes they'll need 10 years of safety data before they're going to approve something.
Yeah, and sort of building on that, I think in the Immuno-Oncology space, we have some really effective therapies like the checkpoint inhibitors, Keytruda, Opdivo, and I think if we use mRNA as a platform for antibody replacement, I think the clinical trial design and the hurdle for that is going to be higher. You're going to have to benchmark against, by that time, years of data. That becomes standard of care. So the value of mRNA has to be significantly higher to really enable that wholesale change, both from a clinical development perspective and a CMC perspective. Because to Andy's point, and I think Adrian alluded to this as well, a lot of these companies, they have infrastructure that's dedicated towards protein manufacturing, and now shifting towards mRNA, that's not going to happen overnight. So I think a few things need to happen. The use case has to be really a killer application where you're really conferring an advantage in the clinical development setting. So it's not just developing another monoclonal antibody against PD-1 for the sake of developing it, but can you improve the therapeutic index? Can you improve the safety profile? And then from a translational perspective, although it's easier to potentially scale, I think a lot of companies have invested billions of dollars in the manufacturing architecture that they have, and it's a very clear path towards enabling approval. And I think outside of COVID, if there's no clarity about that approval pathway, I don't think companies are going to invest in sort of wholesale change in some of these applications for mRNA.
Perfect. Thank you, Sadik. Maybe just a reminder to our audience, we've got about 12-13 minutes left. I do want to open it up. We've got a very knowledgeable panel, hopefully you can see, so I will open it up to the audience in just a minute. I wanted to come back, Adrian, just one more question. You presented on the approach of Capstan to in vivo cell therapy. What anecdotally we're seeing as well is, of course, there's a strong uptake in ex vivo cell therapy, and to this point, lentivirus has been perhaps the gold standard. It's the focus on stable transformation, but just be keen to get your views and any of the other panelists' views on, I think you touched on it a little bit from a safety profile as well, Adrian, the role of mRNA LNPs in perhaps a transient transformation of those cell populations, and how we should start thinking that transient perhaps is a benefit rather than a weakness when compared against lentivirus, for example.
It's very interesting that one of the most shocking pieces of information from our global translational work at Kite when we developed the Yescarta was to realize actually that patients with very durable response and potential cure 5 years plus had only a very transient expression of the CAR, right, for just a few weeks, a few months. But B cells, normal B cells, were regenerating, coming back, which is wonderful for the patients, right, for survivors. So that started to make us think whether an off-the-shelf approach such as allo may actually work in this type of patients. And that led to Allogene, which is one of the leaders in the field. But then stimulated our thinking that perhaps we need to go one step beyond. Is it possible to be able to express a chimeric antigen receptor for sufficient interval of time at that appropriate level necessary for effector to target ratio that will drive a pharmacological effect and polyfunctionality? And that's the idea behind actually Capstan, for example. And perhaps many of you already know there are a number of publications out there showing that you can do that preclinically. Now the question is, how do you translate that to clinic? And the advantage that I see here is major in terms of being able to address disease indications that have a very high, much higher safety bar. We are talking about autoimmunity, fibrosis, regenerative medicine, and so on and so forth, where actually we could, with a minimal exposure, we could impact really the disease progression. Thank you for the question.
Perfect. Anybody else want to comment on that one? Or? No. Maybe a final question, and then I'll open it up to the audience. Sadik, coming back to you, I mean, you shared the example. We're talking here about mRNA, but I think some of the examples that are being trialed right now is combination therapies of mRNA with PD-1 checkpoint inhibitors. We've seen BioNTech perhaps taking a little bit of both of your approaches there, looking at combination therapies of an ex vivo cell therapy mediated by an mRNA LNP boosted by a personalized cancer vaccine. I mean, what are some of the consequences, and how should we think about, Sadik, this combinatorial therapy multiplexed world and what that means going forwards?
Yeah, it's a really important question. I mean, I think if we step back, like traditional drug development, just the complexity of a small molecule is orders of magnitude simpler than a biologic, which is orders of magnitude simpler than a cell therapy, which is orders of magnitude than a gene-edited therapy. So I think right now, the critical raw materials that we're using to develop these therapies are significantly more complicated than the actual drug product that has historically been developed. So I think it's really having that clear line of sight towards what the final target product profile is, and developing it in a very thoughtful and considerate way, because it's going to get very complicated. I mean, I think already we know that CMC is the biggest bottleneck for cell and gene therapies in terms of translation. Now you're compounding that complexity with different modalities, and then also a lot of the expertise in companies, folks develop expertise in a certain modality, and I don't think there's necessarily that fluidity and flexibility from either CMC or clinical development of taking a biologic and a vaccine and a CAR T all into clinical development. So I think what we need to do as a field is really develop that multidisciplinary understanding in terms of drug product development that currently doesn't exist. But I think in order to enable those combinations, you need to develop that multidisciplinary perspective from both the CMC and clinical development perspective to really accelerate the potential of what these things can do.
Perfect. Thanks, Sadik. Anybody else want to?
I think we're going to see a lot more collaboration happening, where there'll be small biotechs that are collaborating with big pharma. Big pharma has their asset, it might be an antibody-based system, and they want to apply a second-generation mRNA approach. And rather than try and build it internally, they'll actually say, okay, we'll bring the innovation in. And so I think we're into that dynamic environment for the first time, where it will happen. It won't be as complex as it could be to start with, but we're going to see collaboration happening between small biotech, big pharma.
Absolutely. Thank you, Andy. So I've been fortunate to hog the attention of the panel for all of these questions. I've primed the audience a couple of times, so at great risk, I'm going to throw it out to the audience, and hopefully there are some questions out there for the panel, either on what it's going to take for mRNA to enable immuno-oncology, or we had company presentations from both Adrian and Andy. So are there questions in the room? And I hope there are questions in the room. No questions in the room? No takers?
We get to pick and choose.
Yeah, exactly.
Start pulling people out in a second. Look,
there's a question.
We got a question at the back.
Thank you.
Yeah, thank you.
No, it's such a fun microphone. Like, I'd be asking a question if I was in the audience. Just hold the microphone.
Thanks for the great presentations, and I'm always excited to see new modalities making it into therapies and into products. So myself being involved in viral vectors, where obviously the analytics is a real challenge. It sounds as if analytics for mRNA should be much easier. What's your experience? What's going to be expected for QA, for analytics of the final products?
It depends on the RNA. There are many different modalities, and so for the larger self-replicating systems, we're a hybrid. So we've had to. We've had to spend a lot of time developing capillary electrophoresis. To be able to get an electrophoregram of a 16 kb construct has been really challenging. And we worked with SciEx, and we have an assay now. But the backbone of what we use for characterization comes from the viral vector field. It's more of a titer assay in cells, so we'll electroporate in our RNA, and we'll measure the ability of the RNA to replicate. There are antibodies that detect those replication intermediates, and then we can measure what's being expressed, whether it's multiple antigens or it's expressed in the cytoplasm. And so with the combination of those, we can characterize the potency of our larger RNAs. Shorter RNAs don't use that, and they're more, they can say this is what the 5' cap looks like, this is what the poly A tail looks like, and it's this length. It's still not all the same. It is a heterogeneous mix of different lengths of RNA, and the regulators are starting to ask more and more questions about how much of it is full length and functional, and what if the 30% is not, what is it, what does it look like? And so the analytics are starting to have to be developed. But I think the modern tools of molecular biology are there.
Maybe just to expand on that a little bit, I think what's interesting is you've got the combination here of the mRNA drug substance and the formulated drug product, and there's actually quite a lot of understanding, not just the efficacy of that drug substance, but the behavior inside the drug product, the interplay of the mRNA inside the LNP can have an impact on the efficacy. So I think there's both considerations around the sort of biophysical characteristics, but also a lot of work on the bioassays. It's one of the things, certainly in precision nanosystems, we focus on a lot is analytical understanding and the interplay of the drug substance with the drug product. That certainly has, hydrodynamic radius of the LNP can perhaps have a significant impact on where it's delivered to the cell. So I think analytical understanding is one clear area, and I think one of the panelists referred to at the moment, I think it was Andy, fundamentally the drug substance, drug product manufacturing, you're working at much smaller scales. These can be intensified processes down to conceivably less than a day. But it's can the analytics keep track with that? If we're thinking about traditional cell therapies where they may be on a 3 to 4 week release, the analytics are not necessarily on the critical path. Here, the analytics is actually one of the things that needs to catch up with some of the CMC. But thank you for the question. I think we've got time for one more question if it's out there in the audience. We do. There's this gentleman here on the...
I just wanted to ask you, I guess I heard that you also use mRNA for developing vaccine against a classic disease like rabies, right? What is, because rabies has a very established vaccine right now, what will make the mRNA technology, but I think you told is a very small amount of what could be like a lot of doses of it?
No, so the rationale behind us picking rabies is, so we have a novel, highly potent, self-replicating RNA system. And so we need to show the technology is working and it's safe, and how does it work relative to conventional technology, and how is it different? And so rabies is the perfect target. You have a naive patient population. Hopefully, nobody has pre-existing immunity to rabies in the US. You have a commercial vaccine that you can benchmark against. And then we can measure, there's a World Health Organization neutralization assay and a correlative protection that reads out to efficacy. And labs that have GMP qualified assays ship serum and they'll give you an answer immediately. And then there's a slew of data out there from other RNA technologies. And so if we can beat conventional technology, that we fly under the adverse event profile that we saw with COVID. So basically, it's just a little tenderness at the site of injection. We don't get this systemic, type 1 interferon response. If the durability of that immune response lasts for a year or 2 years, that's significantly better than the current commercial vaccine. And then the upside is what's going on in the T-cell responses, and these self-amplifying RNAs, replicating systems, really bounce up the T-cell responses significantly. And we've picked a vector that should do that. And so with all of those readouts, we can say to the world, there's a second-generation technology that is significantly better than the conventional linear mRNA and has an advantage over conventional RNA technology. We're not interested in infectious disease. We'll partner with Big Pharma and develop them, let them develop those systems, and we'll generate the constructs for them with the prototypes and do the early work. Our focus is really to take advantage of the T-cell responses in oncology, and then what else can we express? What cytokines, chemokines? We fully believe eventually we'll be able to express antibodies. Maybe not Keytruda, but novel antibodies that have novel mechanisms of action. So there's a whole slew of things that we have our vision on that aren't big, products, but a diversity and array. And we have this library of vectors with just such a diverse set of biology that we're 21 people. And so we can't, we're at a point where we're breaking at the seams. And once we get the value inflection that hopefully that clinical data will bring, we'll find a way of funding the rest of the programs.
Perfect. Thank you, Andy. So I think we've exactly hit time there. I don't want to give our moderator any more kittens in reorganizing this. So it remains for me to say, look, we wanted to talk to you about how we see mRNA really enabling that next generation of immuno-oncology. I think there are so many shots on goal. It might be that they don't all work, but I think there's some really fascinating science out there. Finally, I'd like to just thank the panel for some very insightful feedback, and hopefully you've enjoyed the session. Thank you.
Thank you.
Thank you.